Animals move around. It's not surprising. They move around a lot, but understanding just how and where animals move is key to so many important questions in conservation biology and in management and beyond. Say this bat is endangered and you want to conserve it. You need to know where it roosts, you need to know where it feeds, you need to know where it migrates. Now say this bat carries diseases that could be transmitted to humans. In that case, you also need to know when the bats get in contact with each other and could transmit that disease, and when they get in contact with humans. All of these questions require a fundamental understanding of where animals are moving. Now today, when we carry phones around in our pocket that tell us, that record our exact GPS location and how many steps we've taken each day, I think we take for granted just how difficult this question is. 100 years ago, if you wanted to know where these animals were moving, you actually had to go outside and follow them, which can be difficult. So things started to change a little bit following World War II and the start of the Cold War, when biologists started to think, hey, people have developed all of these surveillance techniques that are being used to track humans. Maybe we could start to apply this to track animals in their natural environment. And in the early days of these efforts to track animals using remote systems, there was obviously a lot of troubleshooting, a lot of trial and error that had to go on, and some of the initial trials actually at Cloquet Field Station near Duluth in the 50s, people were starting to put radio transmitters on grouse that were so big that some of these grouse only made it about 150 feet before they collapsed. So obviously it was challenging in these early days. So enter John Tester, Don Siniff, and a whole powerhouse of researchers that were working at the Cedar Creek Field Station about 45 minutes north of here. So Tester and colleagues made an important insight that they're biologists, and in this case, if they're trying to apply this technology, a nice strategy might be to bring in an engineer as well and collaborate. So they worked hard to recruit an engineer, Cothran, from the Illinois Wildlife Survey, brought him up to Minnesota, brought him up to the U, and together they worked to push this technology further so that it could be applied to animals. And they could go out to places like Cedar Creek, as shown here, where John Tester is capturing a raccoon. They could fit them with a small transmitting device modeled here by this lovely hand model, this smaller radio collar that would not result in an animal collapsing and dying. And then each of these collars would then transmit a specific radio signal to these antennae on top of 75 to 100-foot towers spread across the Cedar Creek Field Station. These towers then would relay these radio signals back to a really large receiving system. And this was the first fully automated system to track animal movement remotely. And although it was fully automated, it still required a huge team of people at Cedar Creek, not only to build and test the system, but also to feed the signal and time outputs out of this machine into for that time, rather sophisticated computerized system that at the end of the day allows scientists to remotely figure out where animals were moving. There are these individual tagged animals out in the field moving around, and they could sit inside and plot where these animals were moving over their lifespan. So developing this system took years of troubleshooting and these whole teams of interdisciplinary scientists and researchers working together. And from the 60s onward, John Tester and others and colleagues started to apply this technology once they started to get it working to lots of different animals, tagging grouse, raccoons, even frogs. So what kinds of things did they learn? They learned things relevant to questions in game management. So say you want to conserve or manage grouse, and they learned things like in the case of ruffed grouse, you need to also intersperse alder habitat in an area if you want to promote ruffed grouse. If you want to have spruce grouse coexisting in the same area, then you might want to have a mix of jack pine as well. And they could figure this out by putting collars on the grouse and basically asking them, where do you guys like to live? They also participated and pushed forward a range of studies that I think in hindsight are very interesting in that they reflect a lot of the fears and concerns of the Cold War era at the time in the 60s. There were a range of studies looking at the effects of radiation exposure on animal movement. So basically in a number of species, both birds and mammals, they exposed them to radiation and then collared them, released them in the field. And interestingly, for some of the mammals they looked at, those individuals that actually survived the radiation exposure, it didn't really change their movement all that much. So since then, radio telemetry and wildlife telemetry in general has come really far and has gone in all sorts of exciting technological directions. So today you can go out and buy a system to outfit on a bee and figure out where bees go. If you have questions about where pollinators are moving to, where they're coming from, where they're feeding, to help manage pollinator populations. You can also use satellite telemetry to track animals. You don't have to have tower systems anymore. You can relay up to satellites. So some recent studies in deer have used satellite telemetry to, this moves, remotely track where deer are moving with respect to deer diseases. So for instance, a recent study showed that contacts between individual deer that might result in the transmission of chronic wasting disease are surprisingly not the highest during mating season, but actually at the end of the winter in March when areas of cover are more limited. So in order to actually manage diseases like chronic wasting disease, you need to know where deer are and when they are coming into contact with each other. In a final example with respect to human diseases, some recent studies using satellite telemetry in birds have investigated how wild waterfowl move around with respect to thinking about transmission of avian flu strains from captive waterfowl to wild waterfowl back to humans. So interestingly, individual waterfowl can sometimes move 200 to 500 miles a day while migrating, but fortunately for disease transmission, they take breaks. And they take breaks for several days in big ponds, and those individuals that are sick, generally they take breaks long enough that they don't keep moving on. They start to become symptomatic and then therefore don't pass the disease further on. So in order to make predictions about how these diseases move, we need to ask these fundamental questions about how the wildlife that is transmitting them move. So across all of these recent studies, it's interesting to think that none of this would have been possible without the pioneering work done by Jon Tester, Don Siniff, and colleagues at Cedar Creek. In fact, the papers that are coming out today still cite this work from the 60s, which is pretty incredible. So we are honored that both Jon Tester and Don Siniff are in the audience also tonight. I'm not sure where they are with the lights, but so I'll end by recognizing them as well.